Bon. Yes. Okay. Okay. So, hello everyone. Thank you for being here today. So we'll make the presentation in English just to make sure that everybody understands. But if your questions are in French, that's also fine. Unfortunately, Olivier won't be here today. He had another commitment, but with John, we wanted to take some time today to present you the Spatial-Cell-ID project. So basically, the idea behind Spatial-Cell-ID is to study the spatial temporal transcriptome of single cells in their tissue of origin. And this is based on three big modules, one that is imaging, another one that is genomics, and to link all of this data analysis, and we will explain you a bit all of this in detail now. So until quite recently, when people wanted to know which RNAs are expressed in the tissue of interest, what they usually did is maybe dissect this particular tissue. In some cases, they could maybe fact sort a population of cells, then isolate the RNAs, make a sequencing library, sequence it using, for example, next generation sequencing. But then this just gives you the bulk transcriptome of millions of cells, and you have absolutely no idea about the heterogeneity of your tissue and about the unique transcriptome of each single cell. So this is a problem which has been actually solved in the last few years by the development of single cell transcriptomics. And I would call that classical single cell transcriptomics because now I think many labs have actually started to do this kind of studies. So usually what you do, you start with a tissue, you dissociate it in a single cell suspension, then you compartmentalize the cells either in nanowells or in droplets. Then you do a reverse transcription to add barcodes in the droplets. You prepare your sequencing library, you sequence all your read or your DNA, and then you do a data analysis. But obviously, your tissue is actually not at all homogeneous. And so you might have a lot of different cell types in a given tissue and a lot of spatial structure. And this is, I think, a very nice example of a very complex spatial structure. And so what you really want to know is where each single cell that you analyze here in your single cell RNA-seq, for example, actually came from in this very complex tissue. So what you could do during the analysis is some cell clustering that will help you to guess the cell types from which each cell was coming from. Based, for example, on some marker genes, but this is not so easy to do. First, because in many cases you don't know your sample so well, and it will never give you the actual location of each of those cells within this complex tissue. So then what can we do? What we really want to do is actually either be able to directly detect all the RNAs in a single tissue by imaging, for example, or be able to know, based on your single cell RNA-seq analysis, where each of those single cells actually came from. So this is actually the point of spatial transcriptomics. So in a way, spatial transcriptomics is allowing you to bridge between histology and imaging methods, where the tissue is preserved very well, but where usually you can analyze only one or a few RNAs at a time, with RNA sequencing, where you can sequence the full transcriptome of your cell, but you have completely lost its spatial identity. So what is Spatial-Cell-ID? Spatial-Cell-ID is meant to be a national equipment to identify and characterize the transcriptome of each single cell while keeping its spatial position in a big variety of different multicellular organisms. So who is going to benefit from that? Obviously you, because you are here, but ultimately we want to open this to everyone, so it will be really a national platform. So when we built the projects, 67 different research teams from 11 units of Lyon were interested. And we can group them in two groups, the core units, which are the IGFL, RDP, LBMC, and SBRE, which are units that either host equipments or will be most involved in the first developmental phase of the project. And there are also, very importantly, their associated service units, SFR Biosciences and SFR Santé Lyon-Est for the wet lab part, and PSMN and CBP for the informatics infrastructure. But then, of course, all the other units of Lyon and, of course, the associated units will be able to use this platform. We also have academic users and industrial users that are interested by this project and already contacted us. And so the goal is that after the first 3 years of development of the project, anybody could use this platform when they want. So where is this going to happen? Here in Lyon. And actually, as I told you, there are many units that are already interested and involved, either in Gerland, in Lyon-Est, or in Ladois. But the equipments themselves will be located either in Gerland, at the UMS Biosciences, at the PSMN CBP, or at the sequencing facility of the IGFL, or at the SFR Santé Lyon-Est, either in the core location of the SFR or at the SBRE. And so how are we going to do that? So what we propose to do is to actually incorporate the latest developments in spatial transcriptomics, some of which are not even commercialized yet. And we really want to be able to study a large variety of different types of samples, small or large number of transcripts, intact or sorted cells, whole organisms, tissue sections, anything you can think of, basically, and combined all these different experiments with the appropriate data analysis, which is going to be a very important part. Okay, so how are we going to do that? So what we did basically is to look a bit in the literature, how people are doing spatial transcriptomics. There are actually many different ways you can do that, some of which rely on equipments and some of which rely on quite expensive and complicated consumables. So we decided to focus on the ones that require equipment because this is an EquiPEX. And so we identified 3 main ways which seem to be the most simple and easily applicable to different types of samples. And so these 3 ways are either using imaging. So with this imaging method, you can look at hundreds to thousands of RNAs and profile them using in situ methods such as single molecule FISH or other types of single molecule FISH experiments. You can also do genomics. I'm losing my mouse all the time. Genomics here, where you can profile RNAs from thousands to millions of cells at a time and sequence them in parallel. And then there is another method that is kind of in between imaging and genomics and uses both techniques, and this is called spatial sequencing, when we will explain you that later. And then, of course, all of this will be combined with the appropriate data analysis pipelines. Okay, so now we will just explain you briefly a bit what these different how these different methods work. So first, starting with spatial single-cell RNA sequencing. So usually what you want to do, you start with your biological material of interest, which can be embryos, for example, here, or different animals or cells or tissue sections, whatever. What you want to do first is dissociate your cells to get a single cell suspension. Then you might want to enrich your population of interest using FACS sorting, either because you want to enrich the particular population expressing a particular marker, or because you want to enrich viable cells. Then you will prepare your single cell omics library, usually using a droplet-based microfluidic system for the isolation of single cells. Then you will sequence your RNAs using, for example, Illumina next generation sequencing. And finally, a very important part is the data analysis. So as I showed you before, you can do cell type cross-layering to try to identify your different cell types of interest. You might want to do lineage tracing, but actually what we really want to do here is spatial reconstructions. So there are different ways you can do that. For example, you can use machine learning types of method that would use either pre-existing in situ images of important marker genes, or you can also use the assumptions that two cells that are close in space tend to have transcriptomes that are relatively close to each other. And so with these methods, you can actually reconstruct the 3D space of your single cell analysis. And this is an actual example of something that has been done. So this is the real in situ image of the expression of a gene. And this is the reconstruction based on single cell RNA-seq data. And this is a Drosophila embryo.
Okay, so my turn. Thank you very much for being here today. So here I'm going to present you a technique that is based on the single molecule fluorescent in situ hybridization, and for the little name is SMFISH. So the goal here is to visualize the transcriptome of your cells without any dissociation. So to see your mRNA in situ. So I'm just going to explain you what is SMFISH. So here you have your RNA of interest in green here, and you are going to perform an hybridization against this RNA with different probes that are labeled with a dye. And then you take your sample, you go to your favorite microscope, and you take a picture of your favorite RNA. Here at the IGFL, we use a method that I think is not working yet. Nothing is moving, right?
Yeah, it's working now.
Yeah, not on my screen.
You are making my mouse move, so.
Okay. What do you see on the screen? The first.
Okay, I can pass for you if you want.
Yeah, do it. Yeah, now it's moving. Okay, so here at the IGFL, we use a technique that is called single molecule inexpensive fish. With this technique, as you can see, the probe are not labeled with a dye, but they are tagged with a flap sequence, and this flap sequence here in yellow will be recognized with secondary probes. The advantage of this technique is the first advantage is it's a technique that is really cheap, because the secondary probe are cheap to design and to construct. And as you can see, you can increase a bit your signal because the secondary probe are labeled with 2 dye instead of one dye. So next slide. So here a picture of what you can get. So in blue and in red, 2 RNA that are visualized with the single molecule FISH technique. So with this technique, you can visualize 10 different RNA if you have 10 different dye and a good microscope. However, you can combine dyes to detect one specific type of RNA, and you can go up to 30 different type of RNA that you can detect with this technique. So the question here is how to increase the number of detectable RNA that you want to visualize. So the answer is to do consecutive cycles of hybridization and imaging. There are a different type of technique to do that. They are called MERFISH, OSM-FISH, split-fish. So for different reasons, we decide to set up a facility using the MERFISH technique. So right now, I'm going to try to explain you how MERFISH function. So first step is to barcode each RNA of interest. So you are going to give a code to each of your RNA. So if you have a 300 RNA, 300 type of RNA that you want to visualize, you will assign 300 different code to each RNA. So the code that you are going to use are binary codes, so 1 and 0. And here an example of 16-byte code. 16-byte code because you use 16 0 and 1s to design your code. Then you are going to design probes against your RNAs, and you're going to perform a primary hybridization. So in one single experiment, you are going to do 300 hybridization. And yes, so here the example of the RNA number 1, so one type of RNA that you want to detect, and you are going to recognize this RNA with primary probes. Then you have your tissue that is ready for consecutive cycles of hybridization and imaging. So as you can see, your primary probes can be recognized with secondary probes. So if you take RNA number 1 in the first round of hybridization, okay, with the secondary probes, you are going to recognize RNA number 1. In the second round, it's a 0 for the code, so you are not going to recognize RNA number 1. However, in round 3, 4, and 5, you are going to recognize RNA number 1. So next. So just to summarize, so what you do is you perform an hybridization that is recognize one RNA. You take a picture and then you bleach your signal and you repeat this several times. So how many times you repeat this process? You repeat it 16 times if your code is 16-byte code. So you perform hybridization and you take a picture. And at the end of the day, what you have is 16 pictures per section if you go to the confocal. And this 16 picture, now you have to perform a decoding. So you have to generate a slide, a picture that is going to summarize all this picture. And this is a kind of picture that you get. So this is your sample. And if you zoom in one section, you are going to see that RNA number 1, 4, and C are expressed in this region. Then you are going to perform cell segmentation. So the goal here is to assign the correct transcriptome to each cell. So this is a classical segmentation. If you can segment a cell if you label the nuclei of the cell, or if you label, for example, the membrane. So after that, you have access to the transcriptome to each cells, and you are going to perform data analysis. So for example, you can perform a cell clustering. Here, a picture of a cell clustering. So the color represents the cell that shares the same transcriptome. And here you can even combine the MERFISH data with a single cell sequencing data to perform a good clustering, a cell clustering. So this is a kind of picture that you can get with this technique. I'm quite always impressed when I see this picture. So maybe you want to know some numbers. So how long does it take to perform these experiments? So after preparing your sample, so after fixing your sample and after performing the first hybridization, you need 24 hours of successive hybridization and imaging. How many genes you can visualize? So far, they managed to visualize 300 genes on tissue sections, for example. And on cell culture, I think they managed to do 10,000 different transcripts. Next slide. So now I'm going to present you the spatial sequencing technique that is called the slide-seq. So all this work. So first, you do a section of your favorite tissue, can be a section of the brain, a section of a plant, anything you want to analyze. And you take this section and you put it on a slide that is called a spatial transcriptomic slide. So in this slide, you have RNA that are barcoded, that are spatially barcoded. So depending on the position of this RNA on your slide, you have a different barcode. So then these RNA are going to be fused with the RNA of your section. And what you do after that, you take just a picture of your section, and then you are going to generate a library. So you digest your tissue, and you generate a library, RNA library that you are going to sequence. So this library, so each RNA on this library contain a spatial information. Okay, they are all barcoded. They are all spatially barcoded. And then after the sequencing, the last step is to perform a transcriptome alignment. And to perform this transcriptome alignment, you're going to use two information. First of all, the information coming from the sequencing. So each RNA has a spatial information. And the second information that you are going to use is the picture that you have taken before the sequencing. So, and thanks to that, you can reconstruct the spatial transcriptome of your favorite sample. Next slide. So how will Spatial-Cell-ID be organized? So Spatial-Cell-ID will be organized around 4 modules. Next slide. So of course, 3 of these modules will be organized around the 3 technology that Yad and I have just presented you. And we have a 4th module, which is a data analysis module. As you can imagine, module number 1, 2, and 3 will generate a huge amount of data that need to be analyzed. So now we are going to present you all these 4 modules. So the module that is based on single molecule fish. So the goal of this module is to visualize the transcriptome of each cell without any dissociation. Will be localized at the Platinum, which is a microscopy platform of the UMS Bioscience. Now they are called URR. And the budget that we have for this module, it's a bit less than 750,000 euros. We're gonna use this money to buy a microscope, a confocal microscope. We don't know yet which one we are going to buy. We have money also, I think 20K or a little bit more than 20K to build homemade microfluidic apparatus. As I told you, the merfish, for the merfish, you need to do multiple rounds of hybridization. And of course, you're not going to do that manually. So we are going to build a machine around the microscope to do the hybridization. So the machine will do the hybridization and the microscope will take the picture. So as you can imagine, this module will need a lot of development. That's why we are going to hire for 3 years an engineer to help us to build this facility. So if you know any engineer who knows to do a single molecule fish, microscopy and microfluidic, let us know. I don't know if this engineer exists, but if you know someone who knows how to do that, just tell us. If you have question about this module, just send me an email. Two other persons are in charge of this module, Sigolène and Jack Brocard. So Jack Brocard is the head of the Platine facility. He has a lot of knowledge on microscopy. And Sigolène, she is a physicist at the ENS, and she knows a lot about microfluidic. So don't hesitate to contact us if you want to join the adventure, or if you have any question. So the other model that we want to set up is a slide sec. So this facility will be localized at the Primastem and Connectometer platform at the SBRI. The two main equipment that are going to be by are a slide scanner, so to take a picture of your slides, and a confocal spanning disk to gain more resolution if you want to have access to a subcellular localization of your transcriptome. If you have any question about this technology, I'm not familiar with this technology, to be honest, please contact Nathalie Beaujean or Pierre Savatier at the SBRI. You can see the email on this slide.
Actually, I forgot to change this slide. It's not a spinning disk. This has changed. Okay. It's a system for live imaging. And just to add to this, that all the equipments that we are presenting now are just examples. We haven't made any final decisions on the exact brand and model, and this is all still in discussion. For some of them, obviously, there is only one type of equipment that does what we want. So there is not really much choice. But for example, here for the live imaging system, for the confocal microscope, and for some other things I will present you now, we are still discussing what model and brand to choose. So now for the spatial single-cell omics part. So we actually divided that in two modules. One, which I will call the large-scale module, that will be located at the SFR Santé Lyon-Est. So here, as I said before, the idea is to quantify the full transcriptome of single cells, which have been isolated. So we have lost their spatial information. They can come from any type of complex tissues. And then we will use a posteriori bioinformatics methods to reconstruct their spatial location and their temporal stamp. So in fact, the wet lab part of this module is not very complicated because it's just single cell omics that many people have been doing already for a few years. It's more the bioinformatics part that is really something new and complex. So for the module that is located at the SFR Santé Lyon-Est, I call it large scale because the idea is to analyze the transcriptome of a large number of individual cells, usually based on a large spectrum of different fluorophores with very good sensitivity, resolution, and speed. And this is mostly because we are going to buy this fax sorter. So this machine is for sure that we'll buy this one. It's the Aurora cell sorter. So this will be the first machine available in the world, as far as I know. So it's a spectral sorter, meaning that you can, like you don't have lasers, you actually sort with white light. So you can really sort with like very different types of populations. And this will be combined with the Chromium controller from 10x Genomics, which does the single cell encapsulation for single cell omics library preparation. And this machine has actually already been acquired by the Profil Expert platform through EPISA funding. So if you have questions about the spatial single cell omics part, you can contact Sophie Pantalacci or myself, and we'll be happy to maybe direct you to other people that can answer your questions or answer if we can. So then the second part of this module, which is the small scale module, will be located in Gerland at the UAR Biosciences, both at the cytometry facility for the fax sorter and at the sequencing platform of the IGFL. So here the idea is rather to do transcriptome analysis of a smaller number of cells, but trying to really maximize their viability, maybe for cell types that would be a bit more fragile, for example. So here we will have also a fact sorter, but a different one this time. So the idea is to have a fact sorter that would be maybe more tailored for single cell analysis, that could do more gentle dispensing to improve viability. So here the exact brand is actually under discussion, and you should have received an email with a survey to answer, and also an email about demos for different fact sorters. So if you are interested about this type of application, please answer the survey and consider participating in the demo. And you can contact the fact sorting facility of the UAR Biosciences if you want more information. So this will then be combined also with the Chromium controller from 10x Genomics, again for the cell encapsulation and single cell library preparation. The library preparation itself actually will be automated using a liquid handling robot. And then finally, we had also planned to buy a sequencer, which is a MySeq. So this is a more small scale next generation sequencer from Illumina, which has actually already been acquired through funding from the ENS, the IGFL, and the sequencing facility of the IGFL. Okay, so then as you could see, actually a very important part of the project is data analysis, because we are going to generate a lot of very complex and a lot of quantity of data that we need to image with quite advanced methods. So we will have a very nice computational infrastructure located at the data center of the ENS, and that will be managed by the PSMN and the CBP. So this will consist of high performance computing clusters, super powerful. I cannot give you more details about that. You can contact Ralph Everts if you are interested, but basically this is more than all the clusters that are already available for biology at DNS. So there will be a lot of computing power. Also storage space, because this type of data is very heavy in storage, especially images. Then with that, we will also have 4 workstations, 2 dedicated for image analysis with actually some special softwares for image analysis, and 2 workstations for NGS analysis. Those are quite powerful workstations that you can connect to from your own laptop, for example, to do data analysis. Yeah, so I mentioned the softwares for image analysis. And because this is quite complex data analysis with also some types of analysis and data that are quite new and recent, we will also hire a research engineer, a bioinformatics research engineer. So there is 3 years of salary included in the Equipex, but the ENS actually added 5 additional years. So we'll have a total of 8 years of funding for this engineer. So the idea is really to set up a data analysis platform that will provide training and help people to analyze their data. The idea is not to do the analysis for them, though. And the key here is really to combine imaging and single cell omics. And this is something like a competence that is not so easily found in bioinformaticians. So again, if you know someone who could fit that profile, please let us know and direct this person to us.
Okay, it's my turn again. So the budget for the Spatial-Cell-ID project is a little bit less than 4 million euros. And 2.4 million euros will be used to buy equipment, and the rest to hire 2 engineers. We have money also for consumables, because the project it's organized in 2 steps. So it's 8-year project. So the first step it's to do development, and after the 3 years we should have a facility that is running. So 3 years is quite short to develop the technique that we want to do, but we're going to do our best. So by saying that, I just want to say, you guys, that if you want to join us, don't hesitate to contact us. because we will need a lot of skills, skills on physics, mathematics, biology. So we really need you guys.
Yes, we really need you to be part of this project. So if you are interested in these new and cutting edge technologies, if you are looking for a new challenge, you can join us to help us develop the platform. As John said, for the first 3 years, there will be a lot of work to actually set up those methods before we can make them available to the whole community. And so we really need a large variability of skills, from wet lab imaging to genomics to data analysis. So really, I think it's quite vast. You can also submit your projects to the platform. That will be maybe in a second step once things are a bit set up, but we will also need some beta testing projects, let's say, to first see if things are working. We also aim at really like this is really meant to be a collaborative project. So we really want to bring people together in Lyon. So we are hoping to organize some seminars, maybe on a monthly or every 3 months, I don't know, basis. Tell us what you think. Even if for the moment, not so many people are actually doing spatial transcriptomics, but I think it's a good way to already start discussing about our projects, share common interests, share tips about how you do things. So not each people is trying to develop the same thing in their own lab, but we can help each other. We will also have 1 day per year, which will be the Spatial-Cell-ID day. And the member of our scientific advisory board will be also invited during that day. And we'll have invited speakers and nice talks, hopefully once COVID is finished. This will be possible. We are also thinking of creating a mailing list to share information, tips, publications, I don't know. So let us know if you think this is good or if you are already into too many mailing lists. So if you want to ask any question or ask to be in this mailing list, you can contact this shiny new email address, spatial-cell-ID@ens-lyon.fr. We are also going to build a website, still not done, but it's on our to-do list. At some point, it will be there. And so, as we said, we really need you. Some people have already joined us to help us build this project. So, I mean, this is just the classical things that are mandatory, like a comité de pilotage, comité consultatif, all these things. But what is really important is the bottom part here, which are the working groups. So those are the people who are really doing the heavy lifting during the 3 first years of the project, helping us develop the different methods. At the moment, for example, we are brainstorming about the different equipments to buy, what is the best brand, etc. So some people are already part of these working groups, but you can still join if you want to join. Don't hesitate to contact us. As you can see, some working groups at the moment are actually empty. Nobody's taking care of that, like utilisateurs et partenaires socio-économiques or websites. If you like making websites, you can also let us know. But there are also working groups for slide tech, for single cell omics, single molecule fish, and data analysis, which are already quite active, and many things are going on at the moment. So if you want to join us, please let us know. And that's it. Thank you for your attention, and special thank you to all the people who helped us build this project. It was a lot of work last year during the first confinement, but somehow it worked. So we are really happy, and now it's really up to you guys to help us make this.
Après, c'était celle de Marie, elle était déjà pas très concentrée. Oui, sur le tube. Mais...
Okay, good. So I will stop sharing my screen now, and now we are here to answer your questions. So if you want to ask a question, you can raise your hand or you can type in the chat if you don't have a microphone. Otherwise, you can just raise your hand and open your mic.
Hello, Olivier. You can ask your question in French also if you want. Doesn't matter.
So Abdul?
Thank you, guys. Can you hear me? Thank you very much for this massive work. I have a very practical question. I already talked in person with Jonathan, so I would be happy to join one of the tasks that you described earlier yet, so we can talk about that in person. But I have a practical question about how would people kind of approach the initiative. Come and be trained and do their experiments, or would there be someone to run the experiments as like a service-like strategy? How do you guys envision the practicality of the project?
So I think the idea is that both things are possible. Basically, it will work as the already existing facilities work. So if you are already an experienced user, you can just go to the facility and do the experiment yourself. But you can also do that as a service if you want and bring your sample and the people do everything for you. Then of course it will be a bit more expensive. So
maybe I can just add one thing is that it's the 3-year development in the beginning where so there's not going to be any service the first 3 years, right? So you have to co-develop the tool in the first 3 years.
So just to rebound on that, so clearly the experiment is multi-level and very complex, from basically the tissue to the final analysis and putting figures, interpreting data and things like that. So I guess this involves a number of people. So how does that work? So I guess with the service, I imagine people bring the samples, explain what they expect, and I expect a certain kind of report at the end. But with the contribution where you come with your samples, so I imagine that you have to talk to technicians, people who run the machines, people who know how to do fixation for that particular type of experiment, bioinformaticians, people who look at the images and image analysis. So it's a multi-step thing.
yeah, so I think that's kind of the goal of the first 3 years. And what the people in the work groups will do is try to benchmark the experiments as much as possible. I think now we have the opportunity, because we have so many groups working with different types of samples, to try to, you know, share the knowledge and try to find common protocols that maybe work well for all cell types, or maybe have different protocols for different types of cells. And then, you know, it will already give guidelines to people who want to come and do these types of experiments. Like, we have tested this protocol, it works like this, and it's a bit easier, you know, you are not just lost, and if you don't know what to do, you already have some guidelines. That's what we hope to do in the first 3 years.
Maybe I can add one more thing. Is so we're all convinced that spatial transcriptomics is the future, very clearly. But it doesn't mean that you need to shift all your omics project to spatial transcriptomics, right? So because this is indeed more complicated, more interdisciplinary and everything. So of course you have to calibrate your project. So if you want to do this, this will require some commitment from your side. So the old techniques are still valid for many questions. Just to mention that.
We can also give you some advices on the technology that is the best for your sample, for your questions. That's also the goal of spatial transcriptomics. So you don't have, at the end of the day, for the facility, I don't think that you have, I mean, for the developmental part, you need to have some skills. But after the 3 years, you don't need to know how to do spatial transcriptomics to use the facilities. All the skill will be on site. That's the ultimate goal, that you come with your samples and we do the experiment.
Benoît Landrin?
Yeah, thank you for the presentation. I just have a question related to that, actually, on the first 3 years. So you are not going then to try to kind of group or like make some type of samples that are like, you know, one type of plant sample, one type of, I don't know, Drosophila sample or something like that to test before, after, like trying different type of sample? Because I guess it might be hard to find like protocols that are able to match for very different organisms, no?
Yes. That's why we need you guys. We need samples at the end of the day.
So initially, are you going to try then to to set up, okay, let's try for the plants, because that's what we do. Like, let's say I'm doing, like, try to develop the technique on one organ, like the root or something, and then later on try to apply it to more, or just try directly different ones?
I mean, we will just try what you guys want to try. So it's up to you. If you have different sample types that you want to try and you want to take part in the development, then we will try it. So of course, we wish to try as many different cell types and sample types as possible. So I think for the moment, to give you an example, in the single cell omics working group, we have people working with rodents, we have Drosophila, we have plants. What else do we have? We have some tumor cells, other types of insects, I think. So we already have quite a variety, but yeah, the goal is really to try different types of cells, and maybe actually the protocols are not that different. Maybe the tissue dissociation, for example, for single cell methods would be a bit different. And even then, maybe all insects might work the same way, or I don't know, something like that. But then I think many steps are quite common.
Okay, cool. I was just wondering if you know, as time is limited, if it was possible to develop in parallel so many different tissues or organisms. But if it's indeed sharing features, then you can gain some time indeed.
It's depending on how many people will join us. If you,
you have 122 people interested, so that's good.
The first 3 years is gonna be a win-win, because you help us to develop the technique, but at the end of the day, you will be the first one to have your transcriptome, the spatial transcriptome.
There is a question on the chat, I guess only me can see it, so I'm going to read it. Quelle est la vocation du groupe de travail utilisateurs et partenaires socio-économiques? Maybe Olivier, you can answer that one.
Well, I mean, I guess so. Of course, it's in the project. So I missed the first few slides. I don't know a meeting before, so maybe you presented it. But there's some socio-economic partners like Leica and all this Clarence and everything. But this type of technology, it's like in the current Platinum actually. Some of the tools are used for socio-economical partners. So this also needs some management, let's say. So we need to know what we do with these people and what they can bring also to the platform, what type of sample, so that there's no competition between what we do in the academic world and the socioeconomic partners. So all this will have to be dealt with. And so we're looking for someone. So yeah, if you're interested, join in.
Muriel?
Yes. I have a question related for how we can help you. I'm obviously interested in SM-Fish, but as you mentioned, to start with that, we need to develop something for doing the several round of hybridization. And how can we help in that?
So to do that, there are many ways to help. First, it's you need to design the probes. You need to design hundreds of probes. Okay, so you have software to do that. I don't know how to use the software actually. So we need to learn how to design the probes. You need to design secondary probes, etc., etc. You need to design codes. Okay, so you need to design a library of code. So we need mathematicians to design this code, and the code will depend on how many samples, how many RNA you want to see. You need to do the actual experiment, single molecule fish. So we need to test different dyes, etc., etc. We need to take pictures. So if you have skill of taking pictures, you can help, and then you need to analyze the data. So you have many different ways to help us. Just to be a bit more precise on this model, there is two parts on the models. We're going to do Murphy, which is a hardcore part, but we're going to perform also classical single molecule fish. Okay, and the goal here is to use a lot of different dyes that are not available, that are, I think most of them are not been tested for single molecule fish. So I think we are going to have a lot of fun of testing these different dyes, how to separate them when you take the picture, etc., etc. So yeah, you have different ways to help us.
Thank you.
There is a question on the chat. Thanks for the presentation, very interesting. Do you have an idea of the cost of an experiment, price per sample, for each of single molecule fish, single cell RNA-seq or slide-seq approach? I think it's very complicated to answer that.
For Murfish, it's between 5,000 and 10,000, something like that. Because if you want to design all your probes and do everything with a private company, it's 500,000. Okay, 500,000 euros, huge, right? But if you do it by yourself, it's a couple of, I mean, I'm not entering in the detail, but there is some protocols that you can use to design your probes, to label all your probes. So it's between 5,000 and 10,000 per sample for 300 genes, sorry.
Yeah, I think it's roughly the same range for single cell RNA-seq between the cost of the 10x reagents and the sequencing and everything, few thousand euros for sure. I don't know for slide-seq, maybe Nathalie or Pierre, if you want to say something, if you're here, you know.
No, I don't know exactly, but I think it would be in the same range of price. But
okay. Abdou, you have another question, or you forgot to lower your hand?
No, I was waiting to see, since there's not a lot of hands rising, so I take advantage. No, it's in the same line, because I guess the idea, well, the question I had, which is similar to what Muriel already asked. So how does it work? Do we come with our budget to help with the development? Does Spatial-Cell-ID project fund that part of the development? And I think the part that will make people nervous is we know that we spend money for experiments and sometimes it end up not working. So buying reagents for 200 euros and not getting a result is annoying, but buying reagents for 10,000 euros and not getting results might give heart attack to some of us. So how do we handle this kind of risk part?
Well, for some protocols, there are some steps where you can already know if your experiment is going to work. You don't have to go until the end and the sequencing or the imaging, maybe for the Murfish to know. And I mean, at some point, if the protocol has been benchmarked, then you tested it maybe on a small scale, then yeah, I mean, it's a risk. It's like everything you do in the lab.
And I guess also the price won't be the same during the first 3 years and then after the 5 years, right? Because if you help with the co-development, of course, it's not going to be the same price. I mean, it's going to be more like there's no maintenance, nothing. So
yeah, for the development part, we have money to do the development part. Of course, you're not going to come with your samples and pay for the experiment. That doesn't make sense. We pay for that, of course.
So I imagine
there will be some projects that will be selected during the first 3 years to really test the method and get things running. So for that, we have some funding. So it won't be money coming from your grants. But then once it's all set up, then yes, you have to pay with your own grant money.
And I imagine in the beginning is probably, we're not going to go for 300 probes. It's probably starting with 10 or 20 is more reasonable.
We are not reasonable.
Thank you.
No more questions? If you don't know how to raise your hand, you can also write question in the chat or
just speak.
Or just speak, yes.
Okay.
Good, then I guess it was super clear. Anyway, if you have more questions, you can write to the email address that I mentioned. So we recorded this, so we will also make it available somehow for people to watch later. And we will let you know also once we have a website. And don't hesitate to contact us if you want to help. And that's it. Thank you.
Thank you very much.
Thank you. Bye.